Hello everyone, and welcome back to my channel. Thank you for joining us today. We're excited to delve into a fascinating topic at the intersection of plant biology and molecular genetics, chloroplasts and RNA. Chloroplasts are the remarkable organelles within plant cells responsible for photosynthesis, enabling plants to convert sunlight into energy and produce oxygen. In this session, we'll explore how chloroplasts function, their unique genetic material, and the crucial role that RNA plays in their operation and the overall processes of cellular metabolism. Prepare to uncover how these tiny powerhouses contribute not only to the life of a plant but also to the broader ecosystem and our planet's health. Let's get started. Chloroplasts are organelles found in plant cells and some algae. They are responsible for photosynthesis, the process that converts light energy into chemical energy stored in glucose. Chloroplasts contain chlorophyll, the green pigment that captures light energy. RNA, ribonucleic acid, is a molecule essential for various biological roles, particularly in coding, decoding, regulation, and expression of genes. It acts as a messenger between DNA and ribosomes, where proteins are synthesized. RNA can exist in several forms, including messenger RNA, mRNA, transfer RNA, tRNA, and ribosomal RNA, rRNA. Chloroplasts and RNA are interconnected through the process of photosynthesis and protein synthesis. 1. Gene expression in chloroplasts. Chloroplasts contain their own DNA and machinery for protein synthesis. They rely on RNA to transcribe and translate their genes, producing proteins necessary for photosynthesis and other functions. 2. Photosynthesis. The proteins synthesized in chloroplasts include those involved in the light-dependent reactions and the Calvin cycle. These proteins are coded by chloroplast DNA, transcribed into RNA, and then translated into functional proteins. 3. Regulation. RNA molecules, such as specific types of regulatory RNAs, can also influence the expression of genes within chloroplasts, thereby affecting the efficiency of photosynthesis and other metabolic processes. Thus, RNA plays a crucial role in the functioning and regulation of chloroplasts, linking genetic information to the cellular machinery that sustains plant life. Photosynthesis in chloroplasts occurs in two main stages, the light-dependent reactions and the light-independent reactions, Calvin cycle. 1. Light-dependent reactions. Location: thylakoid membranes of the chloroplasts. Process: light absorption. Chlorophyll absorbs sunlight, energizing electrons. Water splitting. Water molecules are split, photolysis, to release oxygen, protons, and electrons. Electron transport chain. Energized electrons move through a series of proteins, releasing energy used to pump protons into the thylakoid lumen, creating a proton gradient. ATP and NADPH production. The flow of protons back into the stroma drives ATP synthase to produce ATP. Electrons reduce NADP+ to form NADPH. 2. Light-independent reactions, Calvin cycle. Location: stroma of the chloroplasts. Process: carbon fixation. Carbon dioxide is fixed into a 5-carbon sugar, ribulose bisphosphate, RuBP, by the enzyme Rubisco, forming a 6-carbon compound that splits into two 3-carbon molecules, 3-phosphoglycerate, 3-PGA. Reduction phase. ATP and NADPH from the light-dependent reactions are used to convert 3-PGA into glyceraldehyde, 3-phosphate, G3P, a sugar precursor. Regeneration of RuBP. Some G3P molecules are used to regenerate RuBP, allowing the cycle to continue, while others can be used to form glucose and other carbohydrates. Overall equation. The general equation for photosynthesis is: photosynthesis is the process by which plants, algae, and some bacteria convert carbon dioxide and water into glucose and oxygen using light energy. In summary, it can be described as 6 molecules of carbon dioxide and 6 molecules of water, in the presence of light energy, produce 1 molecule of glucose and 6 molecules of oxygen. This process ultimately converts light energy into chemical energy stored in glucose, supporting plant growth and providing energy for other organisms in the ecosystem. RNA editing in chloroplasts can occur in response to environmental factors. This process involves the modification of RNA transcripts after they have been synthesized, leading to changes in the nucleotide sequence. Here are some key points regarding RNA editing in chloroplasts and its relationship with environmental factors. 1. Adaptation to stress. Chloroplast RNA editing can be influenced by environmental stresses such as drought, temperature changes, or light intensity. These conditions can affect the expression and editing patterns of chloroplast genes, helping plants adapt to varying environments. 2. Improved protein function. RNA editing can enhance the functionality of proteins involved in photosynthesis and other metabolic pathways, allowing plants to optimize their responses to environmental changes. 3. Regulatory mechanisms. Environmental signals can trigger specific signaling pathways that influence the RNA editing machinery, leading to changes in editing patterns that help the plant adjust to new conditions. 4. Species-specific responses. Different plant species may exhibit varying degrees of RNA editing in response to similar environmental stimuli, reflecting their unique evolutionary adaptations. Overall, RNA editing in chloroplasts plays a crucial role in helping plants respond to and survive in changing environmental conditions. Chloroplasts and RNA, while primarily associated with plant biology, have indirect but significant impacts on the human body. 1. Chloroplasts. Photosynthesis and oxygen production. Chloroplasts in plants are responsible for photosynthesis, which produces oxygen as a byproduct. This oxygen is essential for human respiration. Food supply. Plants, through photosynthesis, convert sunlight into energy stored in carbohydrates. Humans depend on these plants, and the animals that eat them, for food, providing essential nutrients and energy. Medicinal compounds. Many plants produce compounds within chloroplasts that have medicinal properties, which are utilized in pharmaceuticals and natural remedies. 2. RNA. Gene regulation. RNA plays a critical role in the expression of genes in all living organisms, including humans. It is involved in processes like protein synthesis, which is crucial for cell function and health. Genetic engineering. RNA technologies, such as mRNA vaccines, have revolutionized medicine. The COVID-19 vaccines, for instance, use mRNA to instruct cells to produce a protein that triggers an immune response. Understanding diseases. Research on RNA and its functions helps scientists understand various diseases, including cancer and genetic disorders, leading to better diagnostic tools and treatments. In summary, while chloroplasts are specific to plants, their role in producing oxygen and food is vital for human survival. RNA's function in gene expression and its applications in medicine directly affect human health and disease management. Researchers are exploring several innovative ways to use RNA in medicine for humans. Here are some key applications. 1. mRNA vaccines. Mechanism. mRNA vaccines deliver a synthetic messenger RNA that instructs cells to produce a harmless piece of a pathogen, like the spike protein of the SARS-CoV-2 virus. This triggers an immune response. Examples. The Pfizer-BioNTech and Moderna COVID-19 vaccines are prime examples, showcasing the effectiveness and rapid development of mRNA technology. 2. Gene therapy. Approach. RNA can be used to correct or replace faulty genes in genetic disorders. Techniques like RNA interference, RNAi, can silence harmful genes or modify gene expression. Applications. Research is ongoing for conditions like muscular dystrophy, hemophilia, and certain types of cancer. 3. Antisense oligonucleotides. Function. These are short, synthetic strands of RNA or DNA designed to bind to specific mRNA molecules, preventing the production of disease-causing proteins. Use cases. They are being investigated for conditions such as spinal muscular atrophy, SMA, and certain genetic disorders. 4. RNA-based therapeutics. Small RNAs. Researchers are developing small RNA molecules, like tRNA, that can target and degrade specific mRNAs associated with diseases. Potential: these therapies aim to treat various diseases, including cancers and viral infections. 5. Diagnostic tools. RNA biomarkers. Changes in RNA expression levels can serve as biomarkers for various diseases, including cancer. Techniques like RNA sequencing are used for early detection and personalized treatment strategies. 6. RNA nanotechnology. Delivery systems. Researchers are exploring RNA-based nanoparticles for delivering drugs or genetic material to specific cells, enhancing the precision of treatments. These advancements highlight the potential of RNA technologies in revolutionizing medicine, improving treatments, and enhancing disease prevention strategies. We'd love to hear your thoughts and experiences with chloroplasts and RNA. What do you find most fascinating about these topics? Share your insights in the comments below. And if you enjoyed this video, don't forget to like it, share it with your friends, and subscribe for more amazing content. Your support helps us keep creating. Thanks for being part of our community. Seedling Sparkles.